go ahead and get started. It is my pleasure today to introduce Ellen Wagner. She actually has a host of connections to this department, or the people in this department. She started her undergrad at CU Boulder, and then just recently actually got her PhD at UT Austin under Jennifer Maynard. So I'm sure a lot of y'all had an interesting opportunity to speak to her today, but let's sit back and listen to her talk today.
All right. Thank you very much for the introduction. I'm excited to be here today to talk to you about some of the research I've been doing over the last 5 years with Dr. Maynard at the University of Texas, mostly focusing around this idea of engineering and applying next-generation antibody and TCR therapeutics for infectious diseases. So to just kind of motivate, why do we think that targeting infectious disease is a reasonable use of our research effort? What I'm showing here is the disability-adjusted life years. So what this means is basically the years of life lost as compared to the life expectancy due to poor health or premature death. So basically you want this measure to be low. And this is plotted globally, and I've plotted all the different conditions as well. So shown in orange are some of the infectious diseases. So worldwide, infectious disease is, certainly a concern. But what's maybe more striking is when we pull out the highest income countries from the lowest income countries and plot the same conditions here. So in the United States, we're of course a high income country. Here you see cancer showing up in this teal color, cardiovascular disease. Infectious disease is really not much of an issue here. However, in the lowest income countries, infectious disease is actually responsible for 41% of this loss of life. And this is really an issue. So this disability adjusted life years takes into account that dying at age 90 of cancer is maybe not equivalent with that same death at age 5, right? So this is why we think infectious disease is really important. So of course, we have vaccines and small molecules like antibiotics, which can be really effective for most conditions. However, they certainly don't work for everything. So for example, some small molecules and vaccines don't really work on people who are immunocompromised. So for example, if you can't make your own antibodies, a vaccine's not going to work for you. So we're interested in how can we protect these high-risk individuals from some of these common infections. Secondly, there are these sort of hospital-associated infections. So you might hear in the news about some of these antibiotic-resistant bacteria starting to become a huge problem, like methicillin-resistant Staph aureus. So clearly, these are antibiotic-resistant. What are we going to do when we have no antibiotics left? Small molecules are not that great at inhibiting these sort of soluble extracellular proteins. So for example, there's a lot of toxins secreted by bacteria, which can actually be used as bioterrorism agents. So the DoD, of course, is really interested in what we can do to protect ourselves against this. And of course, there's always emerging viruses for which we don't have any cures, things that we've been trying to treat for a long time. So for example, HIV, right? Clearly, what we have right now is not working for some of these cases. So maybe to remind you and think about how we might be able to treat these, we want to go back and kind of do immunology 101. So the question is, how does the body naturally target infectious disease? Because of course, most people are very healthy, and we have a lot of mechanisms in place that make this so. So if you're losing motivation, just remember Miss Frizzle told us this back in that Ralphie's Body episode back in the day. So we have one step of adaptive immunity. It's called humoral immunity. So humors of the body, like Hippocrates first told us about, are basically body fluids. These antibodies are present at high levels in your serum, in your blood. Antibodies are secreted from B cells, technically plasma cells. And the structure of the antibody, I'm going to be mentioning some of these words later, so I just want to make sure you know what I'm talking about. This antibody is a symmetric molecule composed of 2 heavy chains, shown in the darker color, and 2 light chains. These chains are linked with disulfide bonds that hold the domains together. The antigen binding occurs just on the tips here, these CDRs. So basically that stands for complementarity determining region. What that means is there's basically this loop of amino acids that's highly flexible and highly divergent, and which actually confers all the antigen binding specificity. So these antibodies are really good at neutralizing these soluble pathogens. Sometimes that pathogen, which we could really call an antigen is what I should say, can be on the surface of perhaps a larger molecule, a larger pathogen. So for example, this is on the surface of maybe a bacteria. So in this case, the antibodies would attack and attach to that bacteria. In this case, we have many cells in our immune system that recognize these antibodies via the bottom region of the antibody. For example, a macrophage, I think, is maybe the most well-known, right? A macrophage pretty much is like a garbage mouth, and it eats everything. So in this case, the macrophage would be able to engulf this bacteria that's covered with antibodies. The second sort of mechanism of immunity we have is called cellular immunity. So this is mediated by sort of a receptor, maybe looks kind of similar to an antibody. However, it's bound to the surface of a cell. So this in particular is a T cell, and the receptor is, maybe not surprisingly, called a T cell receptor, which I'm just going to say the word TCR. This TCR has a similar structure to the antibody. So here, instead of having a heavy and light chain, we have an alpha chain and a beta chain. And again, those CDR loops are what bind the antigen. So in this case, all of your cells of your body are constantly making lots of proteins and displaying those proteins on the surface. Basically, it's kind of little tags of what is being made inside that protein. So in a normal, healthy cell, these are going to be peptides from your own proteins, and the TCR is not going to be able to recognize this. However, in the case of a cell which is infected, for example, maybe infected with a virus, the cell is now making a lot of viral proteins. These are going to have foreign peptides now shown on the surface of this cell, and that foreign peptide is able to be recognized by this TCR. So that will induce, there's several signaling pathways that occur, but one of these would be basically induction of lysis of this now infected cell. So together, these mechanisms are really powerful, and our question, and so when we're coming back to this sort of set of maybe indications for which small molecules and vaccines just aren't working, in our lab, our thought is that we can maybe address these using antibodies and other molecules of the immune system. So our sort of overall goal here, and what I want to make sure I communicate today, is how can we leverage molecules of the immune system to treat and prevent infectious conditions. So I'm going to start out with an example here. So one thing we're really interested in our lab is pertussis, which is also known, and more commonly known as whooping cough. So of course in the United States we have a pertussis vaccine, but you still might have heard of some of recent outbreaks in 2012. So even with that vaccine coverage, we get sort of cyclic spikes of this infection. But more of the problem is that worldwide, pertussis is still responsible for 1% of deaths in children under the age of 5. So there's clearly better drugs needed here. So the pertussis is caused by infection with the bacterium Bordetella pertussis, which colonizes the lungs and secretes several virulence factors. One of these is called pertussis toxin, and it is the cause of severe symptoms. So this toxin consists of an A and a B subunit. It originally, first the B subunit sort of associates to the surface of a host cell. That's then endocytosed taken inside the cell. The A subunit is able to dissociate, and then it goes on and has some catalytic activity and modulates some G protein coupled receptors inside the body. So the results of this, sort of on a body-wide scale, is this increased white blood cell count called leukocytosis, which can be so severe that it clogs small capillaries, can cause organ failure, and of course, eventually death, particularly in infants. So we think that, so our question is, how can we protect and treat infants who are too young to receive, for example, the pertussis vaccine? So of course, we're an antibody lab, and we think that we can do this by using antibodies. So first, we know that pertussis toxin is maybe an important target. So the question is, how can we isolate antibodies that bind this pertussis toxin? So this is basically done by an immunization. So the toxin is injected into the mice. A mouse will make its own antibodies against the toxin, which are secreted from these B cells here. So you can then isolate the B cells and screen a bunch of them and pick out those which seem to be effective. So this will give you a mouse antibody. This was actually, in this particular case, was done by this group in Japan, actually in 1987. My favorite antibody is 1B7 that I'm going to be talking about today. That's just its name. It binds this single epitope, so that's the binding site on the pertussis toxin. It binds on the A subunit and neutralizes the catalytic activity. Of course, we can't really give mouse antibodies to people, so it kind of causes an immune response. So what we need to do is basically make this antibody a little less immunogenic to actually use as a therapeutic. So this is kind of like Grandma's brownie recipe. She gives me the recipe, and maybe I'm using the same ingredients, but it just kind of tastes different when she makes it and when I make it. So we want to make it just like look more like a human antibody. So here you can see this human antibody. We've only retained basically the very tips of the antibody, which confer that specificity, put it on a human framework. So since this is a chemical engineering audience, I wanted to throw in a couple equations just to show you that we can still do that in a biology lab. So of course, the antibody-antigen binding is driven by this kinetic and reversible equation here. And we have these parameters, the forward and reverse association-dissociation parameters, which sort of control this equilibrium. So a measure that we talk about a lot and has to do with the affinity of this antibody is called KD, the capital KD, not to be confused with the lowercase kd. That's a problem with the literature there. So if you look at this, how this is defined, you see that in the case where the binding is favored, so Ka is going to be large because we're favoring the antibody-antigen complex. This is going to cause a lower Kd. So when I mean low Kd, we're looking at things that are measured about 1 nanomolar. That would be about the order of what we were looking for in a Kd. So this can be measured. The gold standard of measuring this is kind of by using SPR. So SPR stands for surface plasmon resonance. So kind of the technology is we have this chip. It's actually gold. That's why it's literally the gold standard. So it's coupled with, it has a dextran layer on the surface, and some of these are terminated in carboxyls. What this means is we can actually do a tiny bit of chemistry. So every once in a while, we actually do chemistry in our lab as well. Basically, this one coupling reaction is the only thing we know how to do. And so we use EDC and NHS, and we can couple this primary amine to the carboxyl group. So this amine is present on lysine, which is an amino acid which is present on the surface of many proteins. So then you can shine a beam of light at the bottom of this chip, measure the angle at which it reflects, and this angle is going to change depending on how much mass is bound to the chip. So you can sort of get a measure of mass onto the chip, which can help you figure out this equilibrium. So then what you do is you have this chip that's been functionalized with, have our antibody bound to this chip. We then flow a bulk phase over it that has a set concentration of antigen. And over time, allow this to equilibrate. So as you wait longer, maybe we're going to get more antigen bound until eventually, if you waited long enough, you would reach this equilibrium condition. So you end up getting a graph that looks something like this. So this is over time. First, we're binding that antigen. We're letting the antigen bind. We're approaching equilibrium as it's flattening off. Then what you do is you remove that flow of antigen. So maybe you're just flowing buffer now. And you see that this starts to dissociate. So we can actually fit these curves and figure out a Kd using some software. So we've done this for our antibody. And you see that as we change from the mouse version to the human version, it's important to note that we didn't really lose any affinity here. And it seems to be about 0.7 or 1 nanomolar. So if you want to read more about these antibodies, we've published a paper on the development. But I just want to show you some of our more recent baboon results. So baboons are the non-human primate model for pertussis, which is kind of interesting. So basically, Gates Foundation is interested in using this as a passive immunization. So basically, in the developing world, you want to give this an infant a shot that basically has antibodies for some common diseases like HIV or pertussis, protect them for the first couple of months of life when they might not have access to hospitals and the disease is most likely to be severe. So to show this in a baboon model, we first take the baboon on day 2 of life and give it a relatively high dose of antibody. Then 5 weeks later, we challenge them with the bacterium Bordetella pertussis, so sort of supposed to look like a natural challenge might be if this was actually given in an infant. Over the next 6 weeks, they're monitored for weight gain, coughs, as well as white blood cell count. So what I'm showing here is some data looking at the white blood cell count. So again, this is our key clinical readout of severe disease. What I've plotted here is the maximum white blood cell count that we saw in each animal during that period after the challenge where we were sort of monitoring them. So the control animals, so they were not pre-treated with anything, have a high white blood cell count. And the the baboons which were pre-treated with our antibody have a much mitigated disease. So here basically it seems that our antibody was actually able to prevent or at least reduce the severity of disease in these pre-treated baboons. So of course my talk today I build as next generation format, so I should probably mention those now. So basically, in some cases maybe a monoclonal antibody, so that's just a single antibody, might not actually be effective enough. In particular, when we're thinking about infective disease, these are highly mutable, that's their job basically is to evade the immune system. So maybe we need to have some more tricks to employ to better target these. So the first way to do that would be by using an antibody mixture. So of course, the natural immune response is highly polyclonal. Polyclonal means there's many versions in there. So monoclonal is one version, many versions, I guess, many clones. So a couple reasons for this. If you have many antibodies targeting the same thing, it's much less likely that they're able to escape this selective pressure. So basically, you can't really evolve if you're being targeted in so many directions. You're not going to be able to escape those all at once. In addition, these might target different pathways. They might neutralize different sites, different mechanisms, and so if you combine these, maybe you can get an even better therapeutic. The original use of antibodies was actually as this polyclonal serum. So back in the 1890s, the sort of high-tech treatment of the day was to take serum from humans, so from blood, serum, which has the antibodies, purify this, and then give it sort of as a prophylactic for diphtheria. It's still actually used today in a couple of indications. For example, if you're suspected of having rabies, you're given this polyclonal serum, but obviously this is not really what we want to do with QC and some of these issues, it's maybe not an ideal therapeutic. In addition, this polyclonal serum, by definition, has many different antibodies, and maybe only one of these is actually targeting something of interest, so you have to give really large doses. So more recently, it's been shown that you can have a mixture of maybe just 2 or 3 high-affinity antibodies that for the target of interest, and they can mimic in a polyclonal mixture. So I said I had one favorite antibody, 1B7, but we actually have a brother to that antibody called 11E6. So 1B7 neutralizes the catalytic activity of the toxin. 11E6 binds actually 2 sites on the toxin here and neutralizes, so basically prevents that toxin from originally associating with the cellular receptors and getting inside the cell. So, as I just mentioned, we think that since these neutralize different pathways, it might be useful to use these as a mixture. This is an ELISA, basically showing that that toxin can be simultaneously bound by both antibodies. This is important because if we can't simultaneously bind a single toxin, it's probably not going to be effective to use a mixture because there's not really going to be any difference per molecule. So basically, first we coat with a mouse version of the antibody, then we come in with the toxin, then the human version of the antibody and detect the human version. So we sort of only get a signal in the case in which all of this binding is actually able to occur. So that occurs with the full toxin. To do, some of these assays are done using this assay called a CHO cell assay. So of course we can do binding experiments in the lab, and those are important to know, but we want to move more toward maybe a human system, of course. So we can use this cellular assay, maybe one step better than just doing an ELISA in the lab. So CHO cells, also called Chinese hamster ovary cells, are a really commonly used strain in our lab. They're used to produce antibodies, and they're really robust. In a monolayer, they normally grow. When they're healthy, they kind of grow in a monolayer like this. However, when you add this toxin, they have this clustered morphology. So you can kind of see there's sort of these clusters here. And what you can do is you can actually look at these yourself under a microscope, or you can have an undergrad look at these under the microscope and sort of try to quantify or sort of, plus or minus, are you seeing this clustering effect? So what you can do is then incubate the cells with a constant amount of toxin that causes this clustering, then titrate in different amounts of antibody and pay attention to the ratio at which this clustering is neutralized. So that would be basically the neutralizing ratio. So what we want to see is that a low ratio of antibody per toxin means that the antibody was able to neutralize well in this assay. So here I'm plotting that neutralizing ratio. So we can see that our antibodies 1B7 and 11E6 were both able to neutralize. And of course, we're interested in looking at the mixture. So using the same total amount of antibody and kind of mixing these in sort of different ratios, we get this sort of trend that maybe a mixture of 1-to-1, an equimolar mixture of the two, is better than either antibody individually. This is definitely not significant, but it does seem there's a little bit of this trend. I also mentioned there's that polyclonal serum. So we have a polyclonal serum that's enriched for antibodies that bind the pertussis toxin, and this does not neutralize very well in this assay. So now we want to maybe do an even better experiment, which is to look at sort of a similar neutralization, but can we actually use an animal model as a readout? So we're looking at white blood cells. So what we do is we incubate the toxin and the antibody and inject this into the lateral tail vein of 5-week-old female bulb C mice. Over the next 4 days, they're monitored, and finally on day 4, we collect the blood, lyse the red blood cells, stain for CD45, which is a marker of white blood cells, and run them on flow cytometry to basically quantify white blood cell count. So here, PBS, which is just a buffer, doesn't induce a white blood cell count, but injection with just the toxin alone does, as makes sense since that's its effect. If we pre-incubate the toxin with either 1B7 or 11E6, we see significant knockdown of this white blood cell count. I should note that in this assay, we chose a relatively low dose. So basically, this is kind of a tidbit of experimental design here. We want to be able to see a difference between our treatments. So we want to choose our assay in such a way that the conditions allow us to actually differentiate between those. So we don't really want to achieve saturation here. So then if we look at the mixture, you can see that the mixture was significantly better than either antibody individually. So basically, this is quantitative evidence of synergy between these two antibodies. So a couple of takeaways from this. First, we see that the mixture of two antibodies seems to be better than either antibody individually. So that's important, and we want to take advantage of that if we're designing a therapeutic. But secondly, I mentioned we were only doing the baboon studies with the one antibody, 1B7. This is because we come up against some very real sort of economic considerations, which is it's really expensive to develop two antibodies at once, right? And we really have to justify whether that's necessary. So this kind of made us think about, okay, how's a way that we could combine these specificities in maybe one molecule or in a different format that we could maybe get rid of some of those additional costs. So that leads me to my next format, which is a bispecific antibody. So if you haven't heard about these, you should read up on them a little bit. They're kind of a hot topic right now. So basically, a bispecific antibody just binds, instead of binding two copies of the same thing, now each arm binds something differently. So these have kind of unique mechanisms of action, as you can imagine. So one way you could use this is basically what I'm calling OR targeting. It could target antigen A or antigen B, just happens to be kind of either, it's a pretty flexible molecule. You could also say if-then type targeting. So basically, MedImmune actually has an antibody like this. The first arm will bind the surface of the bacteria, sort of an ubiquitously expressed sugar on the surface of the molecule, or the bacteria, kind of localize it to the area of interest. And then the second arm can actually bind the therapeutic target. The therapeutic target is at really low concentrations on the surface of the cell, so it's much harder to target if you were just looking for that single specificity. Finally, the original application was sort of this and targeting. So you could imagine only in the case of simultaneous engagement of the two targets do you actually get some sort of therapeutic effect. So this is what's most commonly used in industry and the approved antibodies right now, which is one arm will target, for example, an antigen on the surface of a cancer cell. The other arm will target part of the T cell receptor complex. And so basically it kind of acts as an adapter molecule, and it'll grab passing T cells that aren't normally specific for that cancer target and activate them now near this cancer. So the challenge with bispecifics is this is not really a natural architecture. This doesn't occur in your body besides some sort of this weird, there's one time it does, kind of weird, but so the challenge is how do we actually make these in a cell? So as I mentioned, the antibody binding specificity only comes from those CDR loops. So the interfaces between the domains are the same, so there's no really driving force for them to pair properly. So if you put them all in a single cell, you get kind of this mishmash, and most of it's trash, and maybe only one of 10 is actually what you care about. So a way to do that is to think of engineering this interface to change the structure to actually encourage the mixture that you want. So this is actually done by Genentech in 1996, but it's actually finally now being more used. So what they did is on one of the domains down here, introduced a large amino acid, so a tryptophan, so a really large, bulky, hydrophobic amino acid. On the cognate interface of the other side, they basically took these same sites and mutated them to smaller residues. So basically changing the topography, so now you have a region that is better able to take advantage of this large bulky residue. So what this does is you're sort of favoring this mispairing here. Of course, this doesn't completely solve the problem. This only kind of fixes the two heavy chain pairing, but you still have the heavy and light chain pairing. And this is still a problem, and there's still tons of papers trying to address this issue. It's much more challenging to do this. One way is to pretty much bypass it by just linking the domains. There's also a couple tricks that I'll talk about right now. So of course, we have our two specificities, 1B7 and 11E6. It seems like it might be really useful to design these as a bispecific antibody. So to do that, we introduced the knob and hole mutations into our two antibodies. These were made in CHO cells, purified, and then mixed in a 1-to-1 ratio. This 1-to-1 ratio is then subjected to a controlled reducing agent. So this agent reduces the disulfide bonds between the two heavy chains, but leaves the heavy and light chain pairing intact. So that's our way of addressing the heavy light chain pairing problem. It'll make a mixture of half antibodies like this. You can then remove the reducing agent and allow them to reassemble. And here's another little thermodynamic thing. So we have kind of our Gibbs free energy is going to be minimized more in the case where we have this mixed bispecific format because of those sort of matched interfaces. So we can do a little bit of monitoring of this using SDSPAGE. So here, the knob antibody looks like this when it's purified separately. So we see this heavy band at about 150 kD. That's the intact antibody. We also see a significant band at 75. That corresponds to the half antibody, which is not surprising since we have that sort of mutation that's kind of disrupting folding here. Or not folding, but disrupting pairing of the two heavy chains. The whole antibody is mostly full-length antibody with only a little bit of half antibody because that whole mutation is less disruptive to the mixture. The mixture does not spontaneously reassemble to form the bispecific. However, that same mixture, which does have that reducing agent added, we actually see that the half antibody has disappeared and we have all full length. Of course, this doesn't really mean it's bispecific, it just means that something happened in the reaction. So I learned that when you're in grad school, it's important to have sort of these cheap assays, which your advisor doesn't care if you do 1,000,000 times, right, before you move on to more expensive and analytic protocols. So that's kind of our go and no-go. So to actually figure out if we have a bispecific, we turn to this mass spec method. So we have a core on campus, and I worked with them to develop this protocol. So first, our antibodies are digested with this enzyme called IDS, which cleaves very specifically below that hinge region. So basically, it cuts out about 50 kilodaltons. We have now 100 kD molecule. It's a little bit smaller and easier to run on the instruments. But the pairing information is retained because we still have both arms present. So if we run our proteins on mass spec, we get a unique molecular weight for each one. So here and here is the corresponding to the antibodies here. And this difference is just due to the different amino acids in those CDR loops, which confer the binding specificity. If we run our bispecific antibody then, now we see a unique molecular weight that's independent of either the green or the blue antibody and is in between those. So that's good evidence that we're actually able to make a bispecific here. You can kind of use this to assess purity as well. So you see a little peak here that is kind of contaminating and corresponds exactly with the 11E6 parent antibody. So we can assess this to be about 90% pure, which is well in line with what's reported in the literature for these mutations. Of course, we want to confirm that it actually still binds after we've done this protocol on it. So here I'm showing another one of those ELISAs, but sort of inverse position. So first I have the B subunit, which only binds 11E6 specificities, then the antibody, then the A subunit, which only binds the 1B7 specificities, and finally a detector. So we can only complete this sandwich and get a signal in the case of the bispecific antibody. So that's good confirmation that this actually worked. So now returning to this assay, we want to see, all right, does the bispecific work as well as the mixture? And it turns out it does. So we were really excited to see this. We have about 10 animals in each group here. So at least at this power level, we're not able to differentiate between the bispecific and the mixture. It could be that maybe if we made our dosing even lower, we would be able to see a difference between these two. But at least in this case, it looks like the bispecific actually worked pretty well. So I want to change gears a little bit. And I've been talking about sort of this pertussis toxin and targeting these sort of soluble proteins. But maybe let's think about another type of infectious disease, maybe a virus. So one potential application for some of our technology would be maybe targeting a virus that's prevalent. So this would be human cytomegalovirus. It infects 50 to 80% of adults. It's a herpes type virus, so that means it's present in your body. It remains latent in your body forever once you've been infected. If you're healthy, it's probably a very mild cold or not even symptomatic. However, for people who are immunocompromised or else infants who are infected congenitally while they're in the womb, it can cause birth defects and can be a significant health burden. So in the United States, we actually spend $4 billion a year on kind of controlling CMV in these high-risk populations. And CMV, for 50 years, has been a #1 priority for a vaccine, and we still can't seem to make a vaccine. So this is a really tricky virus. So what's interesting about CMV is that if you look at people that have been infected with CMV, 80% of their T cells, so remember it's a virus, so it's usually T cells that are going to be mediating immunity, 80% of their T cells are specific for one particular protein. This protein is called PP65 and is present in this tegument region of the virus. It's expressed at really high levels and is really immunodominant. So we have, there's that fire alarm thing. So we have this TCR called RA14. It's a public TCR that binds a particular peptide from this protein, PP65. So a public TCR is basically a TCR that's present in a wide diversity of individuals. So it's actually really interesting to think that two people who are unrelated, we might end up having the same TCR. So that means maybe this TCR is actually something really relevant. And that's one other research avenue we're kind of addressing with this TCR. Another thing is that this TCR was pulled from a patient who had arthritis. However, they did not actively have CMV, and they only had very few different types of TCRs targeting CMV. So it seems like this TCR in itself was able to keep that virus in a latent state. So it seems like maybe it's pretty therapeutically relevant. So we think that a TCR, such as RA14, may be able to prevent CMV infection or maybe reactivation in high-risk patients. So just a quick reminder, this is the TCR shown in orange, this is the peptide, and this is the MHC, which is present on your host cells. So our final kind of special format that we're looking at, and this is more recent, is kind of looking at these TCR-directed formats. So for example, the TCRs are something that's interesting about TCRs is that they're really structurally analogous to antibodies and be engineered using the same methods. So I showed that before, but here's our antibody. TCR basically looks like an arm of an antibody. However, it's normally associated with the surface of a cell. However, you can make a soluble format, so you can basically get rid of what's holding it to the cell, and you can try to make the soluble format. So a challenge with working with TCRs is that they naturally have a really low affinity, so 1 to 100 micromolar, as opposed to an antibody, which I said was about 1 nanomolar, is what we're looking for in terms of affinity there. So this makes them really not that good therapeutic agents without finding some way to improve that affinity. Also, they're really difficult to express in mammalian cells. So I mentioned that antibodies are normally made in this mammalian cells. It makes them look more similar to our own antibodies. TCRs are really difficult to express that way. So it's sort of difficult for industry to be interested in this type of molecule. There's one format currently in clinical trials that uses soluble TCR, kind of shown in this diagram here. So basically, they have that same bispecific molecule, but now one arm is replaced with that soluble TCR. It's called an IMTAC. So we kind of want to address some of these issues and see if we can develop a way to better engineer these TCRs and take into account how can we increase their affinity and maybe how can we increase their expression and get them to express in mammalian cells. So I'm going to walk you through the system we've designed. So this is a type of display system, which is a common way to engineer proteins on the surface of a cell. So first we have basically our plasmid. So that's a piece of circular DNA that's containing just our gene of interest here, as well as some other things I'm not showing. So you can transfect this plasmid into this CHO cell and theoretically get display of this TCR on the surface. So here we have a secretion signal, which makes sure the TCR gets to the surface of the cell. We have the 2 chains shown here, and then the second chain has this transmembrane region. This is what anchors it into the surface of the cell. So we want to use CHO cells because, as I mentioned, we want to actually be able to express these from CHO cells. So it seems like we should do our engineering in the same platform, which we actually want to use later on. So we can actually, it's called staining. We can sort of detect these molecules on the surface using these fluorescent stains. So we have a 2-staining protocol that I'm going to show in all my plots today. So first we have an antibody which detects the beta chain and has this sort of yellow fluorophore on it. We also have this reagent called a tetramer. So it's basically that, if you remember, the peptide MHC is what the TCR binds, and we sort of have 4 copies of these making up this sort of tetramer here, and that's basically antigen staining. So this is actually looking at binding. So we get a graph, something like this. So if we run these on flow cytometry, we sort of assess individual cells as they come through in tiny droplets and measure for both the yellow color and the red color, which I know correlates to, on this axis, display of something on the surface, at least the beta chain, and on this axis, antigen binding. So we see that something's binding here. So we might get sort of a mixed population. These are cells which have not been transfected. That's very common in mammalian systems, do their growth cycle. But we see this population here of interest. So there's a linear relationship between the antigen binding and the display. So this is our sort of positive cells. So first we had to do some optimization to even get this system to express, which is not really surprising. So here I'm showing that same type of graph. So we're looking at display of something on the surface, and then we're looking at antigen binding. So here are a couple controls. So blank cells kind of show up in this quadrant here, and we get a little bit of nonspecific binding with tetramer. Our goal is, of course, to see double positives, so things that are staining at this region. An important control, I mentioned that the antibody binds the beta chain only. So what we need to know is, does the beta chain itself also stain with tetramer? If it does, it's going to be really hard for us to differentiate between just the beta chain and maybe fully functional TCR. So if we look at just the beta chain, we see staining with this antibody, but we don't see any staining with the tetramer. So that's good news, so we know that this is not going to contaminate our results. Then we have a couple of variables that we can tune to see if we get to sort of optimize expression of this TCR on the surface of the cell. So the first thing is using the wild type constant regions. So wild type, I just mean sort of what was naturally in this sequence of the TCR we pulled, as well as some modified constant regions. So there are some published mutations in the literature that can help with stability and expression, and we can test those out to see if that helps in this system. So first thing to notice is that moving from the wild type to the modified, we see a large increase in this population that's showing up positive. So clearly those stabilizing mutations are really important for our system. When we're looking at the order swap, so I showed before the cassette that has the genes for the antibody. It had one first and one in position 2, so we can sort of swap those and change which chain is associated with the cell surface. It doesn't seem to make that much of a difference. However, consistently, we got slightly better results when we had it in this order. So alpha's first, and beta is linked to the transmembrane region. So this is the format I used for all of the future engineering work. So the real power with this type of platform, now we seem to have gotten expressing on the surface, is to actually use this for directed evolution. So basically, if we make the same plasmid, but now we introduce a library of mutations. So for example, maybe in the second region here, I've made some changes, and I do a library, so a million different versions here, right? And then I want to figure out which of those is the best, but I don't really know what it's going to be. So if I transfect these, I'll have a bunch of cells that all have different TCRs on the surface, maybe with just a couple of changes. Stain them as I do before, and I might get a graph that looks something like this. So more of a mixed population here. We see some are good and some are bad, so what we want to do is isolate only those that are good and figure out what they actually were. So we can take this region, it's called drawing a gate, take this gated region here, I can sort those cells, so you pull out, you sort single cell sorting, and we can grow these up again, and we can repeat this process several times to better enrich. We can also sequence these cells from this region and figure out, okay, what were those mutations that actually helped us out? So to do this, we first need to develop our library. So the binding region is determined by these CDRs. So what we want to do is actually see if we can improve the affinity of this TCR by changing these binding regions. So if you do a library, so basically what you want to do is, as much as possible, introduce a bunch of diversity in this binding region. So one of these, I pulled out one of these CDRs, CDR3 beta, that's kind of the name of the region, it's associated with binding. So you can see this purple line here, see how close it is to the peptide. So that's basically showing you contact the peptide. It has sort of a structure that's complementary to the peptide. What we want to do is see if we can change those amino acids and maybe optimize them to get higher affinity. So usually, if you wanted to get really higher affinity, you would probably do this on multiple CDRs, and then at the very end, you would combine all the different variants you found. So for one of these CDRs, it's about 3 to 6 amino acids that are important for contacting the peptide. Of these, we found that in this particular case, 2 of them had really extensive hydrogen bonding networks with the peptide and the MHC. So what it seems is like these are basically anchor residues and are maybe really important for binding. So what we decided to do was to keep those anchor residues, the wild type, the sequence that we found in the structure, and then as much as possible, introduce as much diversity in the surrounding amino acids to change the shape of the backbone. So we did this, so this is done basically using a PCR method, so with using degenerate codons to code this. Our final library size was about 1 × 10⁶. So if you read about libraries, this is a pretty small library. However, for this kind of mammalian display system, this is about what you need to work with in terms of sort of practical limits. So now I'm going to walk you through the sorting that we did on this library. So again, we have display on the x-axis and binding on the y-axis. Here's our blank cells, similar to before, and here's an irrelevant protein, so just cells transfected with something that's not supposed to bind and bind at all. Finally, we have our wild type. So this is sort of the original version. So we see the wild type has this, a good positive population. We're looking for versions of the TCR that at the same display level, so the same axis, x-axis, have higher binding with tetramer. So that's going to show up in the triangular gate that I've drawn on the plot here. What we can then do is repeat this process over 3 rounds. So 3 rounds of sorting the cells and then growing up those you sorted and sorting them again. So basically, each time you do it, you're maybe a little bit more stringent on what you're selecting. So you can see from our original round, most of the variants were not very good, right? Because we sort of introduced mutations which were probably detrimental, but there's maybe a couple variants that showed up and were selected. And then from each round to round, we see about 5 to 10-fold enrichment in the variants of interest. So we're really, really excited to see this last round. We've got quite a few following that positive gate. You can actually see sort of 2 tails on this distribution here. Both of those actually look better than wild type. So what we did then was we took those cells and we pulled until we had 10 unique sequences. And what we want to see is, okay, these variants that I pulled, are these better than what I originally started with, to confirm that this selection actually worked. So a couple of readouts. First, does the variant that we pulled retain specificity? That is, if I use a different peptide, which it's not supposed to bind, does it now just bind non-specifically to that peptide, or is it only specific for what we care about? So the non-specific peptide is shown in gray, and so we see these cells are still displaying, however, we're not getting any antigen staining. So that's good news. We've retained specificity as compared to the green variant here. Then when I'm just showing the same plot, but overlapping with the wild types, the wild type shown in black, we can see that, like I talk about, we have that triangular region, and it does look like we're getting a little bit of shift upward for this variant. To try to quantify this, what I did is I took only those that were positive, so pretty much everything to the right of this region. Those are positively transfected, and divided the tetramer binding signal, basically the red color, by the antibody binding signal. I'm basically just linearizing this population. And then plotting this on this plot, and we get this sort of distribution. So this is sort of the normalized signal here, which we want to be higher to be better. So you see the green population is shifted over from the black population. And then, I know this is a lot of sort of numerical manipulation, but if you take the sort of the midpoint of that peak and plot it, that's what I'm using to compare these different variants to wild type. So wild type shows up down here, and then we have our different variants, which all show up at an increased sort of that median point of that curve. So that means that, at least in this assay, they showed shifted over, and we hope that that means that they actually work better. So we were excited to see that all 10 variants are better than wild type. This isn't always the case in libraries. Sometimes you'll pull out some variants that sort of made it through by a growth bias, but weren't actually better. So right now, I don't really know what the difference is in another format. So for example, I talked earlier about using SPR. So our goal would be to take maybe the top of these variants, do those using SPR, and compare that to the wild type, so I can actually kind of figure out what's the benchmark comparing this wild type to this improved version, and what does this amount of difference actually mean in terms of increasing the affinity. So we're at least happy to show that we can have sort of this. We've sort of shown that we're able to use this cell method to sort these and affinity mature. So that's about the data I have that I want to conclude. So I want to kind of tie it together as well as apply it to what the current state of the industry is. So today, I talked about a couple things. I talked about that we can use antibodies and TCRs in situations where small molecules might not be effective. Secondly, we talked about how alternative formats might be really crucial by their ability to have alternative mechanisms. So we could think about maybe using an antibody mixture for targeting pertussis. We can also show that we get basically the same effect by using a bispecific antibody, and there's surely other ways we could engineer these and apply to different situations. I also talked about how we could, this is sort of at this point, just sort of a proposal, but maybe we could use this sort of TCR redirected format for viral diseases. And finally, I showed that we've been working on developing a platform for engineering TCRs, and we hope that this addresses some of the issues with TCRs, including expression from CHO cells and low affinity. So maybe this will make TCR engineering a little easier in the future. So finally, I just kind of want to tie it all together. So I've been talking about antibody therapeutics, but maybe it's not clear exactly what is the state of this field. So antibodies are really a growing industry. So a couple years ago, numbers are always a couple years behind when you look at these things. It was about $58 billion worldwide were spent on antibodies. This makes up about 50% of the sales on protein therapeutics. So they're really an important industry. On this graph, I'm basically plotting years and then showing how many antibodies were approved in that year, how many novel antibodies were approved each year. So all the first antibodies approved in 1986. Recently, in the last few years, you can see that there's actually a huge increase of the antibodies that are being approved. We're well on track to see, in the last few years, 6 new antibodies approved each year. So this really is kind of a growing industry, and there's a lot of excitement in this area. So there's currently 58 antibodies approved and marketed in the United States. Of these 58, only 4 are currently for infectious conditions. Mostly they're for cancer or autoimmune or inflammatory disorders. So these 4 that are approved are CHemE, which is basically an antibody that protects high-risk infants from getting RSV. RSV is a common infection, a respiratory infection in children. So it's basically this prophylactic approach. There's also 2 antibodies that bind the anthrax toxin. Actually, my advisor helped develop this in grad school, so that's kind of cool to see that approved. So basically, these antibodies are stockpiled by the United States just in case of an emergency, so hopefully they'll never actually have to be used, but DOD, I think, did a lot of funding on this. And then actually, just earlier this year, Merck had an antibody approved for C. difficile. So this is kind of interesting because C. difficile is something that you might normally have in your gut microbiome, and it's not usually a problem. However, if you've been in the hospital and have received a lot of antibiotics for a long period of time, it might sort of disrupt the microbiome, and you get these sort of challenging infections with C. difficile. So this antibody was approved to basically prevent reinfection occurrence in people who have already had a problem with C. difficile infection. So I'm guessing that the next approvals, they'll be kind of expanding the indications to actually use this as maybe a therapeutic earlier on. But we'll see how successful this does in marketing. So there are also some of these new formats in the pipeline as well. So there's actually one bispecific antibody approved in the United States. It's for a cancer indication, and there's quite a few more in clinical trials. There are also 40 antibodies currently in clinical development for infectious diseases. This is including kind of some interesting targets like I mentioned CMV, Staph aureus, in particular sort of methicillin-resistant Staph aureus. HIV, of course, we don't have anything that still can treat HIV or can cure HIV. So there's some antibodies in clinical trials for this as well. Also, just actually about a week and a half ago, this news came out that this first cellular therapy it's not quite approved yet, but the board all recommended approval. So basically, this is sort of an interesting therapy. So what they do, if they have this T cell, it's sort of functionalized with actually an antibody binding domain. So basically, you would take this T cell from a patient, you would do some engineering on it to try to improve it, and then put it back in the patient. So this is really notable because it can actually, in some of these cases that have not been able to be treated by anything we throw at it, this can actually cause a complete cure. I think it's, leukemia. So it's pretty interesting. And this kind of paves the way for maybe some of the TCR therapeutics I talked about. So you could do a similar sort of approach with an engineered TCR. So with that, I want to thank you all for listening to my talk today. I'm from the Maynard lab, and we have a lot of people in our lab. Some of the work I've shown you was done in collaboration with our postdoc, Annie, as well as Alam, who's a new first-year student in our group, and then several undergrads that helped with this work. The pertussis antibodies are funded by Synthetic Biologics and the Bill and Melinda Gates Foundation. And then I've been funded by NSF as well as the Welch Foundation. And this is just a shout out to my favorite activity the whole time I've been in grad school was Cheetahs flag football, CHE, obviously for chemical engineering. And I am the captain, was the captain, and we did make it to the championships a couple years. So that's been a great time. So thank you very much.